Guys, and welcome back to Extension Academy. Let's continue our membrane active solute transport. So, in the previous lecture, we were discussing about ABC. Correct? If you remember, we were talking about ABC transporters. What are ABC? ATP binding cassette, right? So, under which we have talked about the MDR, if you remember, and the second one was another example, CFTR. So, which we couldn't complete in the last lecture, right? So now we'll discuss about the CFTR. So that is which stands for cystic fibrosis transmembrane conductance regulator. Now this is also known as chloride channel in the vertebrates. So अभी से clear हो गया है that even though we have got the ion channels for chloride ion, we also have separate channel in the vertebrates which has got the different function. Okay. Now, if there is a mutant CFTR, okay, it affects the chloride ion channel function, which leads to dysregulation of epithelial fluid transport in the lung, pancreas, and other organs, resulting in cystic fibrosis. Cystic fibrosis is आपका disease condition होती है, ठीक है? Now, in this, what happens is that the patients, yeah,
ma'am,
yes,
your voice is not very. It's like your voice is going very.
as I was saying about the fluid, the cystic fibrosis. Okay, so, so now it's like what happens is that the patient, okay, the cystic fibrosis patient, they are going to have respiratory problems then, because what happens is in the respiratory line, there is always a mucus. As I mentioned in the previous lecture also, when you inhale the oxygen, right? Of course, you're taking the air. Along with the oxygen, the bacteria are also there, correct? So these bacteria will get attached to that mucus. So वहाँ पे जो CDR रहते हैं, उनका काम है to remove all this bacteria. But what happens? Because the dysregulation of the chloride ion channel, the mucus gets thickened over there, or onto which the bacteria gets attached, and then they start growing over there. Because of which, what is happening now? It is not able to remove all those bacteria, unwanted bacteria, right? And because of that, then it becomes narrow and the patient is having trouble in breathing. Okay, so these conditions lead to chronic disability and reduced life expectancy. Okay, clear? This much is clear to everyone? Huh? Okay, so this is how your CFTR looks like. Can you see? So this is a membrane, and you can see again in this also NBD1 and NBD2 is present. So where NBD1 is what? Your nucleotide binding domain, right? So ATP is required. So here also ATP required, here also ATP required. Now when this is present, then only the chloride ions are coming or going out. Okay, this is the just the schematic representation of your CFTR. Now, it is found in the epithelial cells of many organs, including the lungs, liver, pancreas, digestive tract, and the female reproductive tracts of male and female. Normally, the protein allows movement of chloride out of an epithelial cell into the airway surface liquid and mucus. Positively charged sodium ions follow passively, increasing the total electrolyte concentration in the mucus, resulting in the movement of water out of the cell via osmosis. So, iska kya matlab ho raha hai? Simple. There is a membrane, right? Now, if suppose you take Cl minus here, what happens? In order to balance the electrolyte, okay, electrolyte concentration, what happens? Cl minus अगर यहाँ आया है तो आपका Na plus भी आ जाएगा. Okay, now Na plus अगर यहाँ पे आया है, Cl minus है, so what is happening here? The salt is forming. अब जहाँ पे salt यानी कि solute concentration is increasing, so what will happen? The water will also move. Okay, to balance the osmolarity between the two. Against the barrier. Is this much is clear? This portion, everyone? Akshay, it is clear?
Yes.
Okay. Yeah. Because based on this only, we have to talk about, and the related animations which I have put will be understood. Okay, so this is again your domains are there in BD1, BD2 is there. So once ATP binds, what happens? So all these regulations, this will get open now when the ATP comes and binds. So here one ATP, here another ATP. Now when these ATPs are bound together, this regulatory domain will actually pull, okay, because over here it will start getting the phosphorylation. Then chloride ion, this is present over here. This will get open up and go outside, okay. Now, what will happen if there is a mutation? So basically CFTR mutant is the deletion of phenylalanine at the 508, yani ki 508th amino acid position, the phenylalanine has been deleted. Because of this, the CFTR is losing its function. Now, this is how, let us say this is the example. This is normal CFTR channel, which moves chloride ions to the outside of the cell. Now, if it is mutant, what will happen? So this opening of the channel will not get happen, and chloride ions are not able to pass through, because of which the mucus is getting collected over here. And because of that, this bacteria will get stick to it and start growing over there. Okay, now here I have got one video over here to show you. So directly from YouTube, I'll be showing. Okay, guys, are you able to hear the?
A patient with cystic fibrosis is
Yes.
To get the audio?
Yes, ma'am.
Yes. Tanvi, are you able to hear?
Yes, ma'am.
Okay.
susceptible to infections of the lungs. This is due to a mutation that affects a protein on the surface of lung cells. Many different mutations can cause cystic fibrosis, but all of them involve a transmembrane protein called CFTR that functions as a channel for chloride to move in and out of cells. In this case, CFTR is less able to transfer chloride ions out of the cell compared to the functioning protein in people without cystic fibrosis. The channel is, in effect, locked shut. This inability to transfer chloride ions has an effect on the outside of the cell. In people without cystic fibrosis, there is a balance of chloride ions inside and outside lung cells, which maintains the right amount of water and salt ions in the mucus lining the airways. Cilia normally sweep the watery mucus from the lung lining, keeping the surface clean. In patients with cystic fibrosis, chloride ions concentrate inside cells and draw in water by osmosis. With less water, the mucus becomes thicker and cilia can't move. Flow is reduced, providing an environment for bacterial infection. But there are now drugs that can help patients. To find a drug, researchers tested over 200,000 small molecules to see if any would bind to the CFTR protein. Then, for each small molecule that had some binding ability, researchers chemically synthesized different variations of that molecule and tested them for the ability to improve the function of some types of mutant CFTR channels. They identified a drug that, when taken by patients with a certain type of CFTR mutation, binds to the CFTR protein, opening the channel and allowing improved ion transfer. This increases chloride ion flow and maintains a watery, clear mucus, which helps the cilia move more freely and reduces the risk of bacterial infection.
Yeah. Okay. Any doubts? Was it clear enough, like how CFTR is working? Yes, Shruti, is it clear?
Yes, ma'am. Yeah.
I mean that's why, you know, like whatever we study and immediately if you watch any video, then that is going to be really very helpful for better understanding. Okay, so right now what we understood, CFTR function we understood. Okay, and this CFTR is going to come again in future when we are going to talk about the different animal physiology related. Okay, so here the main point was like how CFTR is working. So if you have seen, they were talking about these small molecules, like in order to treat the patients, correct? So these small molecules are actually helping or mimicking the binding of the ATP. So if you have seen, if you remember in the structure, when ATP are bound, then only the upper two domains are getting open up. Okay. Now, because of this mutation, the ATP is not able to bound to the NBD domain, the nucleotide binding domain, and hence when this is not getting bound, the upper domains are not getting open up. So जैसे ही आपका ATP bind करेगा, or the small molecules which mimics the structure of the ATP or the activity of the ATP goes and binds over here and helps in the opening of the upper domains. And जैसे open होगा, आपका chloride ion जा सकता है बाहर, ठीक है? So when chloride ion is going, eventually Na plus will go. So solute concentration is increasing. This will also come, the water will come. Now this water will make sure the mucus is not getting thickened, okay? And it will keep on beating so that the area remains clean, okay? Now when this is not working, water is not coming and mucus is getting collected over there. It gets thickened, and to which the bacteria binds. Okay, and will grow hone lag jayega. And of course, see, when it becomes totally thickened mucus and bacteria is growing, then the respiratory problems are increasing. Okay, so that's how the cystic fibrosis is happening. Okay, so this is clear. We can move ahead.
Ma'am, one more thing. Does the sodium pass through this channel or does it pass through its own channel?
Its own channel, obviously, na, because this is specific for chloride ion.
Yeah.
Yeah, so it
Yeah.
will,
So it uses ATP to transfer sodium so that the chloride ion solute balance can happen?
No, it doesn't use ATP over here. It is, see, on your membrane, you have got CFTR, okay? For your CFS. Now, when this is happening, the osmolarity is changing, right? Extracellular, which is resulting in the opening of your Na plus ion channels. Okay, ये passive transporters hai, if you remember in the starting of the lectures we have talked about.
Yeah, but sodium is already, like, there is more sodium outside the cell already, right?
No, see,
So
I got your point. No, it's not always, Shruti. The thing is, you have to always see the scenario, like which particular cell we are talking about. Okay. Since we are talking about here the cell, lungs ka cell. So there the excess concentration of Na plus is going to be different.
Okay.
Outside Na plus nahi hoga. It will be there. But if suppose this is happening and the concentration of Cl minus is increasing, not able to be supported by Na plus, जो बाहर है आपका, ठीक है? Then there might be opening of the channel. So what my point is, see, as you are saying, this is cell, यहाँ पे Na plus का concentration ज़्यादा है, ठीक है? Now, depending on the requirement, we got your Cl minus also. Okay, this is getting and making sure the water is also coming. But again, eventually, at the end of the day, I need to maintain this concentration also outside. Correct? The Na plus का concentration. See, Na plus Cl minus आपका salt बन जाएगा. Okay? The salt and the water is making sure the mucus is getting removed. Mucus क्या रहेगा? आप एकदम slimy, thick रहता है, right? So when you add Na water, it becomes normal watery type. So that gets removed. तो वहां पे क्या हुआ मेरा Na भी कम हो गया. So again I need Na plus.
Okay. हाँ. Yeah, I got it.
Hello?
Yes, ma'am, got it. Clear now.
Now, if suppose the question is coming overall your body, how the body is getting Na plus or Cl minus, then the obvious answer is from your food what you take. Okay, so that's how entire cell tissue is maintaining the concentration of your Na plus Cl minus or the rest of the ions. Now, what are the consequences? If you see, so this is the normally. So what we see, normal airway wall is there, thin layer of mucus is there. So the oxygen supply is easy. Now, what happens if the mucus is there? It becomes narrow. Now, once it is narrow, then the passage for the oxygen breathing and all becomes difficult. And that's why it is difficult for them to breathe. Okay. Yeah. Now, the another topic is bacteriorhodopsin. Have you heard of this rhodopsin? कहाँ पे हमने सुना होगा? Anyone? Rhodopsin. So it is bacterial.
It's light dependent.
हाँ, correct. It is light dependent protein, and this is also the example of your GPCR. ठीक है? We'll come to the cell signaling. There you will talk. There we will talk about the GPCR. Now GPCR. Okay, so let's talk about the bacteriorhodopsin. यहाँ पे ये क्या काम करता है? So this is found in Archaea, Halobacteria as 2D crystalline 0.5 micrometer wide patches known as the purple membrane. ठीक है? So this looks like differently. एक ऐसी crystalline जैसे आपका bacteria रहेगा. Okay, and on that these patches it will be like present. So whenever a light-driven integral membrane proton pump with a lambda max of 568 nanometer containing a bundle of 7 hydrophobic helical rods and an attached chromophore having the carotenoid derivative of retinol, vitamin A aldehyde. Oh, wow. So अब धीरे धीरे जाते हैं. So bacterial rhodopsin is your integral membrane. It's a proton pump, यानी कि ये H+ पे depend है. ठीक है? It is light driven. So यानी कि क्या, जब आपके ऊपर light गिरती है, that is we give in the energy packet, photons we say, right? So that is H nu. H nu मतलब क्या हुआ यहाँ पे आपका? What is H? थोड़ा बहुत फिजिक्स पढ़ा होगा.
Constant.
हाँ, which constant? Repeat one again. Once again.
Planck constant.
It is Planck's constant, and nu is your frequency. Okay. Now, so it is based. So when light is falling, there is a concentration of the H+. Okay, proton का आपका concentration gradient बनेगा. Now with a lambda max of 568. So what does this mean? Lambda max. This is also for the wavelength, right? So we say as lambda max. So what happens is that, before I proceed, and let me tell you. So here we give the wavelength. Okay. Let us say this is the wavelength, lambda in nanometer we represent. Here is your absorption frequency or whatever we give. Okay. Now when a light is falling or any particular substance is present, it is absorbing. There is a particular wavelength at which the substance is going to absorb maximum. Okay. Now if I draw it like this and I mark it like this, here it is given as 568. Okay, 568 nanometer. So this is the lambda max, ये point जहां पे आपका सबसे ज्यादा absorption आएगा. Okay, so that is known as your lambda max. Clear? Is this much is clear? Okay. Now containing a bundle of 7 hydrophobic helical rods. So those are your alpha helices. Attach chromophore. So chromophore for the light or for the color, having the carotenoid derivative retinol. Okay, so this is vitamin A aldehyde. So what we will see is that when this retinol is there, light is पे गिरती है, तो it changes its configuration. ठीक है? Cis trans configuration change होता है, and that is when the rhodopsin gets activated. Okay, we'll come to that. Don't worry. So it acts as a proton pump which captures light energy and uses it to move protons across the membrane out of the cell. So it takes H+, it takes energy, light energy, opens up the rhodopsin and pumps out the H+ out of the cell, okay, which will result in the proton gradient and is subsequently converted into chemical energy. So what chemical energy they are talking about? Maybe in the synthesizing new substance, right? कोई covalent bond बना होगा. So उसमें ये chemical energy आपकी use हो जाएगी. So this is direct example of converting your light energy into chemical energy. So this is a figure here of your bacteriorhodopsin model. The side view, you can see that there are 7 helices are present. Correct? 1, 2, 3, 4, 5, 6, 7 कहाँ है? पीछे होगा I guess. 1, 2, 3, 4. हाँ, 5 and 6 and 7. Yes. You can see over here, the top view, 1, 2, 3, 4, 5, 6, 7 are there. Correct? Now, what do we see the other? So there are in between lipid molecules are there, right? And in here between chlorides are there. Now this is there. Now you can see the chromophore is also present. So you have got the chromophore present over here. Okay. This much is clear. This is just the arrangement. And what do we see? थोड़ा सा gap आपको दिखेगा. This gap is always required so that they can pump out. So the retinal molecule is covalently attached to lysine 216 in the chromophore when absorbs light. Okay, so what does it mean? To the chromophore, the retinal molecule is attached. Lysine 216. 216 is the amino acid position. Okay, now the double bond between 13 changes its conformation from a trans to cis configuration. So this is what I was telling you. So here you can see. So when normally when there is the absence of light, the configuration over here. Okay, here you can see the configuration shows it is trans. Can you see trans मतलब आपका ऐसे ऐसे जाएगा. Okay. Now, when light falls it and it gets open up, what is happening here? The channel is getting open up and it goes into the cis retinol configuration. Can you see the difference in the structure? Yes. Can you see the difference in the structure, guys?
Yes, ma'am.
Akshaya, able to follow? Hello.
Yes, ma'am.
Okay. Yeah, so the concept is clear, right? Like how from trans it is going to cis retinal position, right? Okay. This is important, guys. Okay, this slide is really very important. So at the end of this, a proton is donated to aspartic acid 85 and the proton moves to the periplasmic space. Reprotonation happens by the aspartic 96 restore its original form. Okay, see guys, so when I say that it is getting open up, so what is happening exactly is that the aspartic acid, okay, this is giving its H to the extracellular side. Okay, and this H plus, see यहाँ से H plus आपका aspartic acid 96 है, 96 से आपका यहाँ गया, यहाँ से aspartic acid 85 में आया, 85 में इसको दिया है. So this is how they have restored it. Got the point? Should I repeat? I'll repeat. See, whenever we are saying that the bacteriorhodopsin is getting open up and it is transferring H plus, हमने ऐसे बोला है, right? Exactly क्या हो रहा है? जो आपका इस साइड है, एक्स्ट्रासेलुलर साइड में जो आपका एस्पेटिक एसिड है, ठीक है, इसने अपना H आउटसाइड दिया है. सो नाउ दिस इज COO माइनस. ये माइनस को बैलेंस करने के लिए यहाँ से, ठीक है, लाइसिन के उसमें से आपने H ले लिया है. सो नाउ दिस N इज फ्री. To balance this H has come and joined over here. अब ये हो गया था आपका CO माइनस. इसको बैलेंस करने के लिए इसने साइटोप्लाज्म से H लिया है. So at the end, what is happening? One H has been transported from inside to outside. In short, clear? समझा?
Yes, ma'am.
Okay, this is really important, guys, with respect to your exam. Okay, so finally it is acting as a proton pump which captures light energy and uses its uses to move protons across the membrane out of the cell, and the resulting proton gradient is converted into chemical energy. Okay. Okay. So chemical energy in the form of synthesizing ATP. Can you see? So ये आपका bacteriorhodopsin हो गया, where it is taking these H plus, giving out, this is the outside. Okay, now this H plus is a proton gradient बनाया, और ये proton gradient they have used in the ATPase. And if you remember the ATPase we have talked about, so once this proton gradient is a H plus moves because of the rotation by the gamma. If you remember the gamma rotor which is present in the ATPase, that is converting it into the chemical energy. Chemical energy मतलब क्या? यहाँ पे आपने bond synthesize किया है, ठीक है? Is this much is clear? The function of bacteriorhodopsin. Now here I would like to suggest you that in the case of this, okay, bacteriorhodopsin, and another one was aquaporin. Okay, if you remember aquaporin also we have talked about it. If possible, try to make short notes on the structure of these transporters. Okay, and इसमें कौन से important motives हैं, even though I have mentioned in the aquaporins and bacteriorhodopsin, like for example aquaporin we have talked about NPA. Please talk about please remember that how this NPA is helping in the transport of water, right? And similarly, in the case of the bacteriorhodopsin, just now we talked about the lysine and the aspartic residues. 85, 86 is there, lysine 216 is there. Make a short note on this, make a separate short notes. Either you can make it from Lellinger also, that is will be better. Once we start biochemistry, anyways, I'll be taking, but if suppose it's a repetition, I will be skipping them. Okay. Because Lellinger, cell bio, so much of repetition will be there. So whatever is being covered during the cell bio session, I won't be taking in the Lellinger session then. Okay, that's why I'm saying just make a short notes on this, because exam has come already. The questions have come already from these two topics, the structure and how the motives are helping in the transports. Okay. This is just another example, like in the bacteria, how it is helping. Same thing, the bacteriorhodopsin is present, H plus जा रहा है आपका और ATP से आपका ATP synthesize हो रहा है. So we here in the structures of the bacteria, you can see these bacteriorhodopsin is present in on the plasma membrane here like this. Okay, yeah, clear guys? The time is getting over. Any more doubts are there? Okay, so the next topic is lactose permease. So lactose permease, कहाँ पे सुना हुआ लग रहा है? Have you heard about lactose permease anywhere? Anytime? I'll give you the hints. Mole bio topic में. I have not taken yet, but what about your college में लिया होगा, I'm sure. Another hint, operon.
Lacoperon.
Yes, in the lacoperon. Yes. So there we you must have talked about lac, Z, X, Y, I. ये सारे enzymes के नाम हैं. The genes which operon is getting activated. So there are 3 enzymes which gets activated. Beta galactosidase होगा, एक आपका lactose permease होगा, एक दूसरा क्या है? हाँ, translocase. Yes, correct. Very good. Okay. Yeah. So now we are going to talk about the lactose permease. So even though it's a enzyme, but it is also a transporter. Okay. Okay. So let's talk about the lactose permease. It is actually a symporter. Okay. So Tanvi, can you tell me what is a symporter is?
Ma'am, that transport in both the direction.
No. It transports both in the same direction. Not in the both direction. ठीक है, both direction क्या होगा आपका antiporter हुआ ऐसा. This is both direction. Symporter means 2 molecules in the same direction. So this is symporter. Okay, uniporter means only 1 molecule at a time in one direction. Okay. So it uses a proton gradient towards the cell to transport beta galactosides such as lactose in the same direction into the cell. So इसने पहली बोल दिया, it is your lactose along with the H plus, which is going in the same direction into the cell. Okay, it is made up of 12 alpha helices, which is encoded by the lac-Y gene in the lac-operon. Now the sugar lies in the hydrophilic core of the protein, which is accessible from the periplasm. So कौन से sugar की बात कर रहे हैं हम लोग यहाँ पे? Lactose की बात कर रहे हैं, ठीक है? Which is present in the hydrophilic core of the protein, which is accessible from the periplasm. Now, the binding, on binding, there is a conformational change, which will make the sugar binding site accessible from the cytoplasm. I'll show you the structure and it will be more clear. Now, what is the mechanism? How this lactose permease is happen? It's responsible for the transport of H plus as well as lactose. So the mechanism says that H plus ions from the outside of the cell. Okay, let us make the cell. This is your H plus. it binds to the carboxyl group of the enzyme that allows it to undergo a conformational change. तो यानी कि जो आपका ट्रांस मेम्ब्रेन प्रोटीन बैक्टीरिया है और आपका परमेज ऐसा है, तो सी टर्मिनल यहाँ हो गया, एन टर्मिनल यहाँ हो गया, ठीक है. सो दिस H plus goes and binds to the C terminal. Okay, then there is a conformational change. So this form of lactose permease can bind to the lactose from the outside. Now, when there is a conformational change, the lactose comes and binds to the lactose permease. The enzyme then everts and lactose is transported inward. Okay, I'll show you the structure. Okay, so now here we see. So you have got the lactose permease. So what is happening? The H plus is there. It goes and binds to the C terminal of the lactose permease. What happens? Then the lactose which is present outside will be binding to the lactose and it will be taken inside. Okay. So together what is happening? The lactose as well as the H plus is coming inside. अब बाहर H plus का concentration ज़्यादा हो जाएगा. So then we have got the proton pump. So proton pump will take it back. Now, if you want to inhibit the proton pump, you can use the cyanide. Okay, so now you know the reason why the cyanide is able to kill the bacteria. Right? अगर आपसे कोई पूछे कि how cyanide is helping in killing the bacteria, so you can talk about this, that cyanide can inhibit the proton pump in the bacteria, and hence when there is no proton pumping happening, the many functions like ATP synthesis, the lactose transporter, everything is getting terminated, because of which the bacteria eventually will die. Okay. Now, what about humans? Shruti, can you comment on that? Like, what will happen if humans are taking cyanide?
One will die.
Okay, good. They will die. Okay. Akshaya, why you think they will die? Humans will die?
It may inhibit the neurons,
Neurons.
nerve impulse transmission.
Again, how?
Inhibits complex 4.
It inhibits?
Nerve impulse transmission.
Okay. Shruti, you were saying something?
It inhibits complex 4.
It will inhibit complex core. I'm not getting, your voice is little, I mean, it's very soft. I'm not able to hear it. What is it?
Is it better now?
Yes, yes.
It inhibits complex 4.
Okay, yeah. So see, same thing. When I said over here, the bacteria is inhibiting the proton pump, in the same way here also, anything which is related to the H plus or the pumps, it is going to inhibit. Okay. So see, even if you say ATP also, ATP synthase, okay, ATP synthase, sorry, even that gets inhibited. जहां पे भी आपका H plus related pump रहेगा, it will get inhibited over there. Very simple. जहां पे भी H plus involved है. Now you know that whenever we are breathing, whenever I'm talking anything, within, for every movement, okay, for every involuntary reaction also, like for example in our body, what are involuntary? Heartbeat, blinking of eyes, breathing. This is like not in our control. This is like without knowing also, we are just continuing, right? I am breathing without, you know, मुझे अपने बॉडी को याद नहीं दिलाना पड़ता कि हाँ मुझे ब्रीथ करना है, मुझे ब्लिंक करते हुए याद नहीं दिलाना पड़ता है, राइट? सो ऑल दिस इनवॉलंटरी रिएक्शंस आर टेकिंग द एटीपी. अब जस्ट इमेजिन विद इन अ फ्रैक्शन ऑफ सेकंड योर बॉडी का सारा आप एटीपी ब्लॉक कर दोगे तो क्या होगा? डेफिनेटली यू आर गोना डाई, राइट? सो दैट इज द सडन कॉज. ओके, सडन डेथ के लिए साइनाइड बेटर रहता है. ओके, बट या सो ऑब्वियसली दूर रहना है साइनाइड से. ओके, लेट्स टॉक अबाउट द मैकेनिज्म नाउ. Okay, yeah. So here you can see. So in this also they have shown the C terminal is there. Okay, of your यहाँ पे the pump. Okay, lactose permease. H plus is coming from the outside. It goes and binds. Now when this binding is there, what will happen? Then the lactose will bind over here, and the conformation change is happening. So when this conformation change is happening, the lactose is coming out. Okay, then your H plus is also coming out. Out in a sense matlab out from here, but it is coming inside of the cell. Lactose as well as H plus is coming inside of the cell from the outside. Is the figure clear? See, once the previous figures were clear to you, ये भी बहुत simple है, इसमें कुछ ज़्यादा अभी सोचना भी नहीं पड़ रहा है. What you need to remember is that पहले H plus है, फिर lactose है, ठीक है? Then there is a conformational change, then there is a movement of lactose, पहले removal of lactose and then H plus, then going back to the normal state of the lactose permease. Clear? Yes, Anvi, clear है ये आपको? Do you want me to repeat, Tanvi?
No, ma'am.
Okay. Okay, yeah. So that's all, guys. छोटा सा ही topic था ये. So this we'll be doing tomorrow. ये तो बहुत बड़ा है. Anyways, हाँ. So any more doubts over here in this topic? I guess no. I'll be stopping here now then.